Hello friends, in this video we will talk about the different types of proteins found in the muscles. So we'll be talking about the muscle proteins, and later we will talk about the sarcotubular system, the different components of sarcotubular system. So first of all we will take the muscle proteins. So muscle proteins, there are different types of contractile proteins present in the muscle fibers. So first type of protein which is found in the muscle is called as the myosin protein. Now these myosin proteins, they are found in case of the thick filaments. So thick filaments have these type of proteins called as the myosin proteins. Now if we see a myosin protein, each myosin protein is formed of two long chains. It is formed of two long chains, just like this, two long chains and four short chains. Now these two long chains, a part of these long chains forms the tail. So this entire portion here, this entire portion is called as the tail. This is tail, and the remaining portion is globular in nature, and this portion forms the head. So this portion forms the head. Head is the globular part. Head is the globular part. This is globular part, while as the tail portion represents the fibrous part. Now in the globular part of the myosin protein, in the globular part of the long chains are present the light chains. There are present light chains around the globular heads of the long chains. So if we cut this portion, near the head portion there is greater density, therefore this region is also called as heavy Myosin, or it is also written as HMM, while as tail region is called as light Myosin, or it is also written as LMM. Please note that heavy Myosin is formed of both heavy chains as well as light chains. It is formed of this heavy Myosin. This will be formed of both heavy chains. In fact, the globular heads of heavy chains, this is formed of the globular heads of heavy chains, plus it is also formed of, plus there are present the light chains. It is also formed of light polypeptide chains, while as this light Myosin is formed of only, it is formed of only the heavy chains. It's formed of only heavy chains. The fibrous part of heavy chains forms the light Myosin. Now together if we take this myosin as a whole, so myosin is actually a hexamer. So we will say that myosin is a hexamer because it is formed of 6 polypeptide chains. Is formed of 6 polypeptide chains. These include 2 heavy chains, 2 heavy chains. Heavy chains are those chains which have more number of amino acids, and it is formed of, plus there are present 4 light chains. Light chains are those which have comparatively fewer amino acid residues. Please note that the globular head of the myosin, this globular head of the myosin, it has also enzymatic activity. Globular heads of myosin, globular heads of myosin have enzymatic activity as well. So they act as the ATPase, enzymatic activity, because they act as ATPase. ATPase is an enzyme that will liberate energy by hydrolysis of ATP. So this will cause the conversion of ATP into ADP with the release of energy. Number one. Number second, because these globular heads, they emerge from these myosin proteins, therefore they are also called as cross arms. So the globular heads of the myosin, they form the cross arms. These form the cross arms. So cross arms are also formed of the globular heads. So we can write here that they also form the cross arm. Now a myosin protein or the thick filament is formed of several such myosin proteins. Several myosin proteins will bind together and they will form the thick filament. Now from the thick filament, these cross arms will emerge from the surface just like this. So these are the cross arms or the myosin heads that are emerging from the surface of the thick filament. Now these bundles of myosin proteins are held together within this thick filament by means of a protein which is called as M-line. So M-line is responsible for binding the different number of myosin proteins together. Now these myosin proteins or the thick filaments, they are also in contact with the Z-line. Z-line and there is present a protein which connects the myosin proteins with the Z-line. That protein is called as the titin protein. Now this titin is a non-contractile protein but plays an important role in keeping the myosin proteins or in keeping the thick filaments at their specific location. So this protein which connects the thick filaments with the Z-line, this is called as the titin protein. Titin protein, you should remember, this is the largest protein of our body. So it is titin protein is the largest protein of our body. It's the largest protein of our body and it is present between the Z-line and the thick filament or the myosin proteins of the thick filament. So this was about the myosin protein. Another type of protein found in muscle is the actin protein. So as far as this actin protein is concerned, actin protein is found in case of the thin filament. So this is present in the thin filaments. This is found in thin filaments. So as far as this actin protein is concerned, actin proteins, they are the globular proteins. It is a globular protein. It is present in the form of globules, and the globular form of the actin protein is called as the G-actin. So globular units of the actin protein are called as G-actin. G stands for the globular. So these are the globular actin. And several globular actins, several units of globular actin, they will polymerize and form a fibrous structure which will be called as the G-actin. So let me draw a G-actin. Suppose this is one globule, then several globules, they will polymerize together just like this. They will polymerize and form a long chain and form a chain-like structure just like this. This chain-like structure formed of several G-actins is called as the F-actin. This will be called as the F-actin. Suppose what I am drawing here is the F-actin. So F-actin and G-actin, they are actually F-actin is formed by the polymerization of the G-actin. So here is this will be called as now F-actin. F stands for fibrous actin. Fibrous actin. Fibrous actin. Apart from this actin protein, there are other proteins present in the thin filament. Thin filament is not formed of actin protein alone. There are present other proteins in it. For example, they are very small proteins with respect to mass, but they play an important role in the regulation of the muscle contraction. These proteins include number one, these other proteins present in thin filament. They include tropomyosin. Now this tropomyosin, this is actually a fibrous protein. It is a fibrous protein. It is fiber like, and its job is to cover the actin sites during the relaxed state. So when muscle is in the relaxed state, this tropomyosin will cover its sites. So its job is that it covers the actin sites during the relaxed state. So when the muscle is in relaxed state, when muscle is not contracting, at that time the tropomyosin will cover the actin sites, those actin sites which will bind with the thick filaments or the myosin proteins. Next type of protein which is found in case of the thin filaments is called as the troponin. Now this troponin, this is a globular protein. It is a globular protein and it has 3 units. It is formed of 3 units. A globular protein formed of 3 units. Now the 3 units of the globular protein includes number 1. First type of globular protein of troponin is called as TNT, or it is also written as TPT, troponin T or TNT. This troponin T, it is a globular protein, a component of the globular this troponin, which is attached to the tropomyosin. So its job is that it is attached to the tropomyosin. Number 1. Number second type of second unit of the troponin, that is called as TNI. TNI is actually the inhibitory protein. So it inhibits the actin protein during the relaxed state. So it is an inhibitory protein and it inhibits the actin. So its job is to inhibit the actin during relaxed state. And number third and final unit of troponin is called as the TNC. TNC is that component of troponin which binds with the calcium. So it is the calcium binding unit of the troponin. Now as far as their distribution within this thin filament is concerned, they are present along with the actin filaments. For example, this tropomyosin, that is present in the groove that is present in the F-actin. So F-actin, as you can see, within the F-actin there is present a groove, and this groove, in this groove is present this tropomyosin just like this. So tropomyosin is found in the groove of the actin filament, as well as there are present these troponin units at the regular intervals. For example, here I will draw now the troponin. So troponins are formed of 3 units. I will draw them in different colors. So suppose these are the 3 units of the troponin which are present at different places, right, just like this. So here one, here will be another one, so on and so forth, and here will be another one. So these are the 3 units of the actin. So this was about the types of protein is found in the muscle fiber. So we have seen that the number of the type of protein is present in these thin and thick filaments. They include myosin protein. Myosin protein, we have seen that it is exclusively present in the thick filament alone, and we have seen that it is a hexamer. It is formed of 6 polypeptide chains, 2 heavy and 4 light chains. It has a tail and a head. Head is globular and it has the ATPase activity. It forms the cross arms, while as the tail, it is fibrous in the nature. We have also seen that the bundles of these myosin proteins are held together by M line, and the thick filament is in contact with the Z line by means of the titin protein. Then we have seen that in muscle fiber there is another type of protein that is called as actin protein. Actin protein, we have seen that it is present in the thin filament. It exists as a globular protein which is called as G-actin, and those G-actins will polymerize and form the fibrous structure called as F-actin. Apart from this, there are present 2 additional proteins which are regulatory in nature. They are smaller in number quantity wise, but they are regulatory in nature. These include tropomyosin, which is a fibrous protein, and its job is to cover the actin sites during the relaxed state. Next we have seen the troponin. Troponin, we have seen, is a globular protein which is formed of 3 units, and these 3 units include TNT, TNI and TNC. TNT attaches to the tropomyosin. TNI is a inhibitory protein which inhibits the actin during relaxed state, and TNC is the calcium binding protein. Now we will talk about the next part of this video, that is the sarcotubular system. So let us talk about now the sarcotubular system. Sarcotubular system is actually formed of 2 components. So first, sarcotubular system. Sarcotubular system is formed of 2 components. The first component is called as T-tubule, and second component of it is called as sarcoplasmic reticulum. Now what is this T-tubule? T-tubule, if we again observe, if we will show diagrammatically, the sarcolemma, which is a plasma membrane of a muscle fiber, that is drawn inwards at regular intervals at some places, and those invaginations are called as the T-tubules. The invaginations of the sarcolemma that innervate into the sarcoplasm are called as the T-tubules, because they resemble the letter T. As you can see, these resemble the letter T, therefore they are called as T-tubules. Now these T-tubules, we can say, what are these T-tubules? These T-tubules are the invaginations of sarcolemma, and they are present at the AI junction. AI junction is the junction that is formed between the A band and I band. At their junction are present these T-tubules. So sarcolemma will invaginate into sarcoplasm at the junction between dark band and the light band, especially in case of skeletal muscles. In case of cardiac muscles, they are present at the Z lines. So that is the first component, that is T-tubule. So T-tubule is simply the invagination of sarcolemma that innervates the sarcoplasm. Now second is the sarcoplasmic reticulum. We can write it as SR. Now SR or sarcoplasmic reticulum, these are the cisternae of the sarcoplasmic reticulum are present between these T-tubules. In other words, each T-tubule will be flanked by the cisternae of the sarcoplasmic reticulum. So these sarcoplasmic reticulum, these are the storehouse of the calcium. So their function is that they store the calcium of the sarcoplasm in them. So they are the storehouses of calcium of a muscle fiber. So these are the cisternae of the sarcoplasmic reticulum plus the T-tubules. They together form what is called as sarcotubular system. So this is the cisterna of SR. SR stands for Sarcoplasmic Reticulum. Now, what happens during muscle contraction? A muscle will contract only when the motor neuron, which is present at the neuromuscular junction, releases certain neurotransmitter like acetylcholine. Suppose here I will draw a motor neuron. So motor neuron will end at axon terminal, just like this, and that axon terminal will form a synapse with the sarcolemma. So this junction between the axon terminal, so here it is the axon terminal, this is motor neuron, this is the motor neuron, and this is here the sarcolemma, and this is sarcolemma. The junction between two, the junction formed by motor neuron, axon terminal of the motor neuron, and sarcolemma, this is called as neuromuscular junction. So here I will label it. This is the neuromuscular junction. Neuromuscular junction. At the neuromuscular junction, this motor neuron will release the chemicals or the neurotransmitters, just like this. It will release neurotransmitter like it releases acetyl coenzyme. So acetylcholine. This acetylcholine will then bind with its receptor located on the sarcolemma. So sarcolemma will be having the receptors for binding with the acetylcholine neurotransmitter. So once this acetylcholine binds with this receptor, it initiates the depolarization, it initiates the muscle contraction, and then muscle will contract, which we will discuss in the next video. Hope you have understood this topic today. So in next video, we will talk about the mechanism of the muscle contraction. Thank you.